Góðan dag og velkomin á Málstofu Hafrannsóknastofnunar. Þetta er fyrsti fyrirlestur núna á vormisseri. Til að nýta tækifærið og hvetja ykkur ef þið hafið einhver erindi, endilega hafa samband. Í dag fáum við erindi frá Lorraine Valliant. Hún er sem sagt doktorsnemi við líf- og umhverfisvísindasvið og leiðbeinandi hennar er Guðbjörg Ásta sem er sviðsstjóri hérna á Hafrannsóknastofnun. En Lorraine hefur sem sagt aðsetur við Rannsóknasetur Háskóla Íslands á Vestfjörðum og hefur unnið við rannsóknir á vistfræði hafs og vatna og sviði stjórnunar strandsvæða. Rannsóknarverkefnin í doktorsnáminu snýr að því að skoða samhengi fars, atferlis og vistnotkunar hjá ungviði þorsks, ufsa og hjá gönguseiðum bleikju og urriða. Í dag ætlar hún hins vegar að tala um mastersverkefnis sem hún gerði líka fyrir vestan og fjallar um þéttleika ungviðis þorsktegunda og kóralþörungi á Íslandi. Gjörðu svo vel.
Hey. Hi everyone. Thank you so much for joining me today on my lunch lecture. So through the introduction, as you know, this is based off of my master's thesis done in 2019. And additionally, we have collected more sampling in a few years after that. So the title is Juvenile Gadoid Abundance on Maerl Rhodolith Beds in Iceland. So to start off, not a lot of people know what maerl really is. And so just to give a brief background about that, maerl beds are calcified algae that grow as rough textured branch-like formations, and the term maerl refers to various species based on how they calcify themselves. So maerl beds are found from the water surface to 100 meters in depth, with the highest abundance between 20 to 30 meters deep. Here's just a very small video just to show you what that can look like underwater, because not a lot of people know what maerl beds can really look like at that depth. They can grow for very long periods of time, and studies have stated that the regrowth rate is so slow that it can be considered as a non-renewable resource. It has been reported in northern Icelandic fjords that maerl habitat is noted as widely distributed and can consist of a wide diversity of epifauna and fauna species. It has then been documented that fish are utilizing the habitat as nursery grounds, and particularly important commercial juvenile gadoids. So as gadoids transition from their pelagic life stage, most depend on shallow nearshore waters, nearshore areas. There is no specific preference pre-settlement, but post-settlement stages, as they enter into the benthic areas, they prefer structurally complex habitats. So this transition of choice is usually for territory and gain of refuge, as well as predation protection. It has also been stated that nursery grounds are defined from high population densities of juveniles, where the growth rates and survival rates are high to provide a recruitment supply to the adulthood populations. It has been reported that maerl was found in areas such as Arnarfjörður and Hvammsfjörður, as well as Kláfjörður, where maerl is located on exposed rock faces within these fjords. From 2011, there was geological surveying of maerl thickness and distribution in Arnarfjörður, Ísafjörðardjúp, and Jökulfjörður of Iceland, where there was a claim of about 170 million cubic meters of calcified algae in Ísafjörðardjúp and Jökulfjörður. The map illustration is from a published article from the Icelandic Museum of Natural History, where Ísafjörður became of interest for me to conduct my master's thesis in 2019. I'm now going to go over a few examples of anthropogenic impact on coastal environments in the West Fjords. There are several, but just to give a couple examples. The first example is that there is an interest in dead maerl extraction. And so maerl is considered a traditional harvesting in Europe, as its skeleton is made of calcium carbonate, so chalk, and is used as a soil conditioner to treat acid contamination. In the 1970s, France has used maerl as an animal food additive, where extraction reached about 600,000 tons of maerl per year. And then in Brittany, other uses were for water purification, mineralization, and cosmetics. So there is a company, a lime algae company, that's currently conducting dead maerl extraction in Arnarfjörður, and they hope to expand into Ísafjörður. And so this is the map illustration here, highlighted in green, is areas where they're proposing to do their extractions in Ísafjörður. And this is also then based off of the geological surveys that had been conducted in 2011. So the second example is that there is interest to expand fish farms in Ísafjörður from various companies, some close to where the maerl lime company projects hopes to expand for extraction. There are lots of past studies that looked at the effects of fish farm cages to the surrounding marine habitats. Many of us are very aware of this through the literature over time. An example of a study could be from the Bay of Brest in Scotland, where there was up to a 100-meter radius of impact to the surrounding maerl habitat due to an increase of debris and decrease in biodiversity. So overall, these two types of anthropogenic impacts to coastal environments can contribute to the cause of maerl habitat fragmentation. So specifically for the presented study that I'm going to talk to you about today, I'm going to show you where the study locations were chosen based off of articles that are coming out that are about maerl distribution within Ísafjörður. So the areas here and here in the red boxes are where the locations are done. So in Hestfjörður and then in Ísafjörður. So for the study objectives, so the presented study, the first is to examine the role of maerl beds for juvenile gadoids at the time of benthic settlements. And then the second is to compare dive and camera surveys as a tool to estimate the juvenile gadoid use of maerl beds. So specifically how the dive surveys of fish abundance and camera surveys conducted on maerl beds to then compare with the adjacent adjacent sandy gravel habitats known as like the reference sites. The surveys then took place in 2019 as well as in 2021 from the onset of juvenile settlement into the benthic habitat, and over the following weeks from between July to the end of September. So this is just an illustration to show you an example of what the dive surveys had looked like. So we followed scuba survey methodologies based from coral reef monitoring efforts and eelgrass bed studies. This illustration is specifically from the reference of real-life survey, as this was the original photo. The transect lines were 50 meters long, placed perpendicular from shore, and the species of fish were counted within 10-meter intervals over the transect line. And the overall dimensions then end up being 5 meter by 5 meter and then 10-meter intervals of the line. So from there, the surveys then started from the deeper end of the transect line and moved into the shallow end. So for camera survey methods, I did use a GoPro footage during my surveys while I was scuba diving to help with ground truthing of the substrate height, as well as the use for any fish species that may have been missed identified. From there, to compare the dive surveys with underwater video footage, I placed a stationary camera at the shallow end of the transect line to record while I did the survey starting at the deeper end. The camera footage of the fish count was then compared to the fish count from the diver of the first transect interval. And this was done in Hestfjörður only of 2021 for the 3 transect lines that were placed within that study area, and for 5 dives where the camera recordings were then compared. So this is a map illustration of where each transect line was located, so between Hesfjordur and Isafjordur. The smaller figures represent the transect lines with the 10-meter intervals by 5 meter of either side of the line. The estimated maerl percent coverage per each interval is also shown here. That's how the lines are highlighted in different colorations here to give off the estimation of coverage. And as you can see here, there's 3 lines in Hesfjordur and then 5 lines in Isafjordur with 2 reference sites. I then created a sediment classification scheme based on past publications so that we could record the composition of different substrate types and for the estimated maerl percent coverage for each transect line interval. On the left are photo examples of what maerl patches could look like during the dives. They're not to scale, but I just wanted to give you guys that visual of what it could look like while I was diving in these areas. So the figure A is an example of maerl percent coverage along the transect line that could have coarse sand and silts. B is an example of majority of maerl with some silts. And C is an example with little to no maerl against cobble, boulder, and gravel. This is an overview of all the dive surveys, where there was 48 dives that were done in total and extended throughout the summer season. A total of 238 individual juvenile gadoids were counted, most often juvenile cod, but also safe. So it was 153 to 36 respectively. All the juvenile gadoids were most likely zero group individuals, where they're about 40 to 100 millimeters in standard length. Other fish species observed were the European plaice, rock gunnel, juvenile flatfish, and lastly, a large sole of zero group juvenile herring was observed transversing the maerl beds. So for the statistical analysis side of the surveys, there were 2 generalized linear mixed models used to estimate the effect of maerl percent cover and environmental variables on, first, the number of juvenile gadoids observed in each interval, and second, the presence or absence of juvenile gadoids in each interval as well. The predicted variables were either the juvenile number or presence of absence for each interval, and the fixed effects for the model were then the maerl percent cover class, visibility, depth, sea state, tide, month stated as a factor, and year. For the transect ID, this was considered a random effect to account for the potential spatial autocorrelation of the intervals within transect. And the juvenile counts were modeled with a negative binomial distribution, where it was excluding the 0 counts, and presence and absence with a binomial distribution. The model fit was then examined and confirmed to meet assumptions of residual normality using the package called D-HARMA. So this is the output of the GLLM tests. First, showing the estimated effects of maerl percent cover and other environmental variables on the number of juvenile gadoids, and 2, the presence of or absence of juvenile gadoids for each transect interval. So there was significance at 60 to 80% maerl percent cover, where it was more frequent to see juvenile gadoids based on abundance and absence versus presence. There was no significance of other fixed factors of the depth, temperature, visibility, sea state, and tide level. In 2021, there was a higher frequency of seeing juveniles, which is likely explained because of the camera surveys. And through August and September, they showed significance of seeing juveniles over time, where I'll now go into explaining that a little bit more further. So the number of juveniles observed was highest in July, and this was explained by shoal size rather than by the frequency of observation. This is reflected in sightings of larger juvenile aggregations in July, followed by more frequent individual sightings or smaller aggregations in late August and September. This pattern coincides with behavioral changes from juvenile cod pelagic shoaling to defending territories. solitary behavior, and also dispersal from patchiness as the fish increase in size. The smaller juveniles were observed in maerl thalli and agree with maerl acting as shelter, and overall suggesting that the diverse and structural rugosity may be particularly important for habitat transitions of zero group Atlantic cod. Maerl percent cover was the only factor significantly explaining the likelihood of observing the juvenile gadoids, and the likelihood was highest in transect intervals with 60 to 80% maerl cover. The reason for this is very unknown, but it is widely distributed in the study, whereas the intervals with 80 to 100% or 40 to 60% were rarer, and the statistical power behind those observations was therefore less. Looking at the raw data can suggest that the number of juveniles was generally higher on maerl beds than on coarse sand or gravel or coarse sand or silts, like at the reference sites. And even using a Kruskal-Wallis rank sum test for juvenile number on maerl transects versus reference transects with no maerl showed high significance. More juvenile gadoids on maerl than on adjacent sites. Our results then support a higher frequency of zero group juveniles within complex habitats. There were significantly more juveniles counted on the stationary underwater camera than by the scuba diver survey at the same site and time. Irrespective of visibility, more fish were observed by the underwater stationary camera than by the divers. The cameras were not baited, as bait could attract large aggregations of fish and aggregation also of just larger fish in general. So camera counts were performed consecutive to the diver's count of the same area. I'm just going to show a brief video, so I'm just going to exit the presentation quickly. There we go. And I just wanted to show you guys a video of what the stationary camera could look like. So the fish are quite small, but you might be able to pick some of them out. So for these underwater, so there's one right here actually. You can see him. So I had to spend lots of hours watching these videos to really count the individuals and know who is who that was coming into view. There's one right here. Yeah, they're very tiny. Yeah. Okay. But yeah, I just wanted to give you guys that perspective of knowing what it really looks like underwater. And here's another one right here, actually, as well. As you can see, my eyes are very trained to pick them out. Yeah. Just seeing if there's any other ones I can point out to you. Yeah, so there's one right here. Anyway, I'm going to continue on with the presentation now. I know you just turned off the lights. I hope that's okay. So, previous studies suggest that juvenile densities post-settlement depend on the complexity of habitat. And I've been saying this throughout the presentation quite frequently so far. And past studies found that survivorship depends on the rugosity and complexity, where the lowest densities were in sandy substrate. And this was by Tupper and Boetleer. For another past study from Lorraine et al., they found that within site temporal variation correlated with the abundance in sand, but not in eelgrass, suggesting that a few large aggregations represent high abundance in sand, and that eelgrass sites are consistent with individuals over time, even though less variable at high abundances. Our current study is comparable to these numbers, where we had densities between 0.16 to 2.56 juveniles at each 10-meter interval intersect per dive. However, the estimate with the camera surveys was much higher than the scuba surveys, with one juvenile per observation on sand per gravel and 5.75 juvenile gadoids per observation on maerl. Visibility was generally low during the study, between 2 to 5 meters, and this could result in fewer observation as the fish could hide or swim out of view in response to the scuba divers' presence. The surveys in Iceland can be affected by low visibility, factoring in with unpredictable weather and current activity, where it is likely to result in lower number of observations. For this presented study, as the divers pursued surveys, it was observed that the juveniles would swim closely to the bottom substrate and have moments of pausing next to the substratum until the diver moved closer. This is similar behavior as to what other studies have looked at for cod juvenile behavior for responses to potential danger. Therefore, the low visibility and juveniles' behavior cause challenges as the fish camouflage with the substrate or swim away from view. Maerl beds have a history of overexploitation and protected in areas along the Atlantic and Mediterranean Sea. The distribution in the North Atlantic is not adequately documented, and there are a few studies on the associated macrofauna overall. So for current knowledge on maerl distribution and density in Iceland, it's mostly based on proposed extraction sites, but there is also other past studies that have been conducted over time. There's no restrictions protected from impacts, neither bottom contacting fishing gear nor direct extraction. And habitats may therefore be at risk of degradation or fragmentation, where predation may be higher and risking survivorship of juvenile fish. So in conclusion, maerl beds are a habitat for the juvenile gadoids, specifically at the time of benthic settlement, and beds with higher maerl percent cover seem to be favored. This then supports previous studies suggesting that post-settlement juvenile gadoids have a preference of complex habitat. And for future studies, we encourage underwater video senses, particularly stationed units over transect line intervals, that can capture the video logs of these zero group juveniles for their absence of presence and behavior without unnecessary aggregation or disturbance. The study then highlights the significance of conserving maerl beds for their functional role as nursery grounds for post-settlement life stage of juvenile gadoids. Here are some acknowledgements, specifically to those who helped a lot when I was doing the dive surveys, especially for safety measures and for the equipment. And thank you so much for listening to my talk today. Here's some information about myself if you'd like to contact me regarding questions about this or my PhD, as well as for the Research Center of the Westfjords, University of Iceland, if you'd like to know more of what we're doing out there.
Thank you, Michelle. Are there any questions? I have one.
Okay.
Now, your last point. You showed us figures of proposed anthropogenic disturbances in Ísafjarðardjúp.
Mm-hmm.
Do you think we know enough about nursery habitats for gadoids or other species to be able to just place them? It looks like it's based on some kind of information, but is it? Do we have enough information?
From my understanding, I think that more is definitely, more information to gather is always good, especially for updating and knowing what's going on based off of past studies or any information that we just continue to still not know. So I think that it's important to have more.
These are calcified structures, right?
Calcified algae.
Yeah.
Yeah.
Sea pens in the Westfjords have been using stuff to kill sea lice.
Mm-hmm.
Is this going to be affected by this substance,
Uh,
these algae?
To be honest, I'm not entirely sure for the sea lice specifically, because I wouldn't know. I don't know what the chemicals are like that they would be using. But just based off of past literature, there is a lot about just debris in general and fecal matter that has cause
And
effects.
fecal matter, are they going to affect the maerl beds, the beach?
I'm not. I don't know what has been done here in Iceland specifically for that, but in other parts of the world in the North Atlantic, they have shown effects.
Nope.
Thanks for a very good presentation. My name is Guðmundur Óskarsson.
Okay.
I was wondering, and I'm sure you showed us, what was the depth of the study area? And second question,
Mm-hmm.
were you studying, were the same depth at all the study areas? I'm just wondering if a depth is an important factor in determining if there is a cut there, or if you should be looking at deeper stations or something like that.
Mm-hmm. Through this study particularly, it wasn't showing significance of depth for differences. Looking at this further, I think that's always good, of course, to continue or to look deeper for sure. The depth that our transect lines were at, they vary between about 8 meters to 20 meters deep. And I chose that also specifically as I was a scuba diver and making sure that I wasn't going too far away from my limits.
Yeah, thank you, Michelle, for a really interesting talk. I just have a question regarding the kind of the comparisons between the still camera work and the dive transect. Could you compare them directly, or was it more that you were comparing kind of the GoPros between the sandy, or like you showed, but could you compare, and how did you compare, the dive transect with in regards to numbers, while the others are stationary, and I guess, briefly, how do you do it? Do you count them in for several minutes, or then you count them again, or is it a cumulative numbers of juveniles you've seen, for a given time slot? I was just curious. I might also look it up in your thesis. Thank you.
Yeah, no problem. So these, for the stationary cameras, this was only done in Hesfjordur in 2021. So there were 5 videos for the 3 transect lines that I did this comparison to, and it was only on the very last transect interval that I did this comparison. So when I would do the dive surveys, I would place the cameras at the shallow end of the transect line, and then would go up, swim over to the deeper end where the end of the transect line would be, dive down, do the survey, and at that time that camera would be recording. And I would time it in such a way of taking a specific interval of the GoPro footage to then compare with the transect interval of only the first interval of when I was diving, like what I counted at that point. Yeah. Does that answer your question?
Yes, almost.
Okay.
It's I think it's always complicated when you're comparing, something which is stationary, but as you described that, then you compare, then you are doing the transect at the same time, and I guess, you estimate, how far you see with the GoPros and all that, but I'm always, it's an interesting thing, to leave the GoPros and let them count for some time, and pick them up, and then again you could deploy them at greater depth that, would not limit, to your dive. Because I guess again, seeing the juvenile gadoids is difficult, and then also, going down with the gear is also quite tricky in many of those areas to catch them, especially in deeper.
Yeah, definitely.
More questions? Já.
it's more of a comment, no? I just wanted to comment on it was really tricky to get the transect lines out with the depth, just to find like comparable habitats in all of these areas at the same depth, because often, of course, in the fjords it's just very steep down. But I think definitely the site selections were as good as it gets in that sense. But I'm actually interested in, because discussing also this with Kalle, and I mean with you describing what you saw during the dives, there seems to be a lot of sort of sediment movement, and often the maerl beds are just quite covered, so making the maerl in itself much less visible. So how permanent are these beds, in your opinion?
How permanent are they?
Yeah, are they just like... I know they have to be quite old. We know that.
Yeah.
But, yeah. Permanent as habitats. Maybe that's more precise.
Yeah. I think that's a main comparison of how thick the layering would end up being for the beds. Like how many of these maerls are just like leveling and lying on top of each other for them to stay in these areas for such long periods of time. I think that's really my only comment, because I'm not entirely sure of how much the current would really take into account of shifting them so much. But that could also be based off of tidal levels, etc.
Sort of moving material so that it's fully covering the maerl, but it would survive, and then it's just exposed again. Just, okay.
Yeah, go for it.
It's just a comment also
Okay.
regarding this, the age of these deposits. They have been measured in the West Fjords, and they're up to 10 meters thick, and their accumulation is thought about 1 millimeter per year, which means that these deposits started being formed just after the Ice Age, 10,000 years ago. Yeah. And I think you get often sediment on the maerl, but there is very much biological activity inside the matrix, where you have animals going in and out of the matrix, turning around these nodules, and the sediment falls down in between.
Yeah.
So you get sediment accumulating under the live maerl, which is only about maybe 5 to 10, 15 centimeters thick, and that's the sediment underneath, which is just like very soft sediment, or mixed with maerl fragments, and that's what they are actually extracting. But again, these can be exposed to lethal sedimentation, which we observed now in Eyjafjörður, where we had an inflow of sand in the shallow sublittoral zone, and we had a live maerl bed underneath. Yeah. But when we came again now, this sand had disappeared from the shallow sublittoral zone, and there was a cover of sand over the maerl bed. So we started taking the sand away and found that whole nodules, not fragments, just whole nodules of maerl dead underneath the sand. So it's vulnerable to too much sedimentation, but I think in a normal situation they can cope with some limited sedimentation by these biogenic or organisms that are moving them around, getting in and out of the maerl.
Yeah, that's really cool.
Yeah, I think they can all hear me. Yeah, okay, online, yeah. I mean, this is interesting because it's key to their function as a habitat. To the larger, well, to the fish and other larger species there.
Yeah, just knowing that there is a high amount of biodiversity of various types of species there. Yeah.
Very interesting, Kalli. Takk. Any more questions? if not, then thank you very much, Michelle.
Okay. Thanks, guys. Yeah. Thank you.